Okay, so Dr. Charles is working in the microbiology department, Faculty of Sciences, Edo State University, there, Edo State, Nigeria, where he utilized the applications of biological techniques and microbial bioprocesses for the actualization of sustainable development goals and agrarian revolution. Through quality teaching, research, and community development, he was formerly the acting director of intellectual property and technology transfer, the head of Department of Microbiology, and sub-dean of the faculty, and is currently the chairman grant committee and the AG Dean of Faculty of Sciences. He is a visiting professor and executive director for the Center of Biotechnology, Precious Cornerstone, University of Ibadan. He is presently an external examiner to many academic institutions around the globe, including Department of Microbiology, University of Namibia. He has won several scientific awards and grants from renowned academic bodies like Council of Scientific and Industrial Research, India, Department of Biotechnology, India, the TWAS, Italy, Netherlands Fellowship, the Agency of International Development Cooperation, Israel, Royal Academy of Engineering, UK, among many others. He has published many scientific journal articles and conferences proceeding in the refereed national and international journals with over 390 manuscripts. He is bioinformatician is working in the post-harvest management and nanotechnology. He was recently appointed as the president and chairman governing council of the Nigerian Bioinformatics and Genomics Network Society. He was recently appointed as the director of International Affiliation and Training Center for Environmental and Public Health Research and Development, Zaria. So I would like to invite Dr. Charles Oluwaseun Adetunji to come online and unmute his mic.
Good morning, everyone.
Good morning, sir. We can hear you. Good morning, sir. We can hear you.
Okay, thank you very much. I'm so happy to come to this great conference. And I want to thank Dr. Shakira for inviting me to give a keynote talk. It's very, very early here in Nigeria. So this morning, I'll be talking about next generation animal gut microbes, recent trends, and their diverse advantage everywhere. So, as we know, the whole world is going through a lot of diverse challenges. So it has become imperative for us as a scientist to find a kind of solution that can help us to solve a diverse problem. Recently, the whole world has gone through the era of COVID-19, and it has become very imperative for us to solve, to look for a lot of solutions that could help in mitigating several challenges facing the whole world. I am a distinguished professor of biotechnology and microbiology. Now, as it has been stated that the whole world is going to increase to 9 billion in the year 2050, the use of gut microorganism or micro have become one of the sustainable tools that will help us in resolving the global problem. And one of the major problems facing the whole world is the issue of sustainable food and how we can provide an employment to diverse part of the world that are going through a lot of challenges. Now, a big problem before us is that gut microbiome serve as a tool that will help in resolving the global problem. Yes. Now, as I stated, one of the 4 major goals of our time is achieving food security. How do we avoid dangerous climate challenges and high rates of Omicron? Now, food microbiome. Let's quickly go to microbiome. Microbiome represents human microbiome biography, which contains of different microbiota in various parts of our body. From the same side of individual, there are different types of microbial composition of different type and from different individuals. Now, intercommunication occurs between the host and the gut host, which shows that gut microbiome are known to communicate with the host in numerous ways through the production of metabolites, modulation of immune system. They could also alter gut permeability, which in turn the host will influence the gut microbiome in a variety of ways, including altering the human environment through diet and immune modulation. Also, going through these 2 statistics, genetic material is extracted from fecal sample, prepared through sequencing. Digesting genetic sequencing are filtered to quality and prepared to database to identify the nature of such microbiome. Now, also, scientific tools and models have been used to investigate various microbiomes, which shows a lot of general overview about interaction between microbiome and the host. Example of techniques currently available in the whole world include human clinical trials, conventional germ free animal studies, interfacial study where animals are given fecal microbiome, anti-pre and probiotic symbiotics, and the general study effect. Now, there are several factors that could influence the composition of microbiome, and a wide variety of factors are influenced, ranging from environmental factor and genetic factor. Now, the effect of different composition of gut microbiome has been stated, which include the Western diets, plant-based diets, and Mediterranean diet could influence the various types of gut microbiome. Microbiome explication of various disorders. Now, most researchers have shown evidence that the role of microbiome play in a lot of disease management, such as skin, metabolic, and cardiovascular disease, as well as cancer, infection, and neurodegenerative and psychological disorder. Now, fecal microbiome transplantation has been used as therapy also, especially from the stool, from a heavy donor collected and screened for safety. I'll be talking about that later. Now, how do you improve your gut microbiome or your gut health? Now, we'll be talking about how do we identify nutrition, lifestyle, and environmental factors that will affect gut microbiome. To be able to define and determine the use of various probiotics and prebiotics, we'll get to apply H principle to diet and lifestyle. Now, microbiome and microbiota shows a lot of richness and variation of bacteria in the gut that could affect good bacteria in the gut. What advantage do they have? They help in mitigating a lot of barrier, help to make vitamin B12, K, folic, improve immune system, provide barrier to bad bacteria, produces short-chain fatty acid, and energy for cell. What affects gut flora? Age could affect, long-time dietary habits, lifestyle, stress, weight, and some medication could also influence the level of microbiota, like antibiotics, antacids, and a lot of other things. Now, what are probiotics? They are living bacteria or yeast that provide a health and commonly used food. BL3 are some of the examples, and you can find out all the examples on the screen. Probiotics function. They promote, restore, reversal of diabases, absorption of vitamins, and strengthen immunity and prevent pathogenic colonization. They could improve digestion and research pending also as some of the advantages that they have. Now, they are used in management of diarrhea. They could serve as chemotraveler, antibiotics associated. Now, they play a lot of role in constipation management and a lot of other diseases like obesity, diabetics, allergy, teeth health, depression, and many other ones that I might not be able to talk about. They also play a key role in dentistry, in allergy, depression, and anxiety. Now, let's move on. So probiotics have come to be a kind of safety that mankind are now banking on because of their numerous advantages. Now, duration also play a lot of role. So probiotic supplements contradiction, hospitalized patients, newborn, immunocompromised patients, cancer immunopatients, hepatic cancer management, antibiotics. So, but it's always good to discuss with your medical doctor to provide the best solution. So probiotic sources like lactobacterium, biobacterium, dairy, and some non-dairy like yogurt, fermented soy, fermented vegetable are also good. Probiotics food yogurt like bacteria, live active culture, non-dairy example, are all many other ones are also very good. Prebiotics are non-digestive substrates that are also good. Greater stability, short chain acid that decrease the pH of the colon and help in the management of H. pylori, help to decrease cholesterol level, may reduce risk of colon cancer. Also symbiotic therapy. Some example of prebiotics are shown on the screen, like onion, garlic, barley, honey, bananas, and a lot of other fortified foods. So not all probiotics are credible equal. Some of them have conditioned and recommended prebiotics that we can use, like diarrhea for antibiotic usage, irritable bowel syndrome, weakening immunity, lactose indigestion, constipation. So you can see some of the strains that could be used in their management. What can we do to improve our gut health? Change your diet, plant-based diet, high-fiber diet, which include diet from varieties of energy and sources, limit excess calorie consumption, limit your sugar, fatty processor, and cut back your caffeine and alcohol, consume probiotics in rich food. Do not overuse antibiotics, manage your stress, sleep enough, exercise. Now, how do we produce a hygiene hypothesis, most especially around microbiome, and how to mitigate a lot of allergy in children? Now, we can increase children allergy by increasing a lot of other practices that has been used. For example, increasing a lot of antibiotics use, decreasing a lot of antibiotic use, breastfeeding, and a lot of other ones could also help. Microbiome of allergic children shows that an overrepresentation of antibacteria and lower proportion of bifidobacteria. So this has a lot of things to do. So antibiotic growth promoter has also come in place because they could stimulate a lot of the use of antibiotics and form a kind of permanent replacement to many antibiotics. So there are a lot of consequences can effect of antibiotics and the microbiome be established, but because of my time, I will not be going through deep of this. So the gut microbiome could serve as a reservoir that could replace a lot of antibiotic resistance that have become to be global challenges. So antibiotics can occur through various methods, and a lot of co-selection of gene has also been experienced, and auto-vector and transmission of this gene could be mitigated or prevented through the use of antibiotics. So replacing antibiotic resistance with pre and probiotics agriculture and aquaculture has been another great part of research that can help us to detect a new strain or use of gut microbiotics. So their efficacy of probiotics can play a lot of role in fish production, in animal production, and in maintenance of human health. So eating of yogurt will play a lot of role in this function without any effect. Now, livestock, how could human guts and its role play a kind of immunity? I have a lot of questions. What type of cell could be found in typical human body? Maybe at birth, after birth. Each human become a unique community made up of our own human cell. Talking about bacteria, fungi, archaea, and a lot of other. So should doctor consider patients a single human organism or community of organisms? It's a very big question we need to answer. Question 2, what is the community of human host and its microbiome called human microbiome? We need to look critically into this. Which do you think is more similar to your microbiome, your classmates' microbiome? A human earth is microbiome located.
Hello, Dr. Charles, are you there?
Okay, am I audible now? So
yes.
Another question is, what are your microbiome organism doing? You need to emphasize on the role and place more emphasis on them, whether they are enemy, foe, or something else. Human, a complex of ecosystem of microbiome that play a lot of role. You can see them on the screen. Now, the gut microbiome immunity play a lot of role, from gut intestine to immune system. They play a lot of role. Now, throughout our lifetime, microbiome secrete compounds that regulate immune system, like the T cells. They are cytokine, chemokines, and a lot of other chemicals. So our microbiome secretes a lot of compounds that may determine our future health. Most essentially, relative proportion of bacteria produce short-chain fatty acids that differ significantly between stool of healthy eggs with human behavior and colorectal cancer. So, which of the most important role of the gut microbiome do we talk about? Another question is microbial gene. There are a number of times more microbial genes are present in the microbiome than our own human gene. We need to play emphasis on this. Microbial gene turn off and turn on responses. Do we recall, maybe the recall lack of brain? We need to focus more on this. Our gene turn on and turn off response in what do microbiome. What information should scientists use from a list of gut microbiome present that we have? A list of major enzyme operating in a gut microbiome, what do you think might change pregnancy? Okay, am I audible? So as our microbiome change, as we come growing with puberty, changes in diet, medical condition, and treatments, might and pet name, and many other factors have been investigated. So also, we need to know and be sure the kind of microbiome changes that occur, the kind of microbiome present, number of each type of micro, relative amounts of microbiomes, and the kind of microbial, active microbial gene. You need to compare most of the recent mean you and your classmate or your peer are taking, predict how many microbes that may different as a result of different mean. What method does scientists use in human microbiome? Many of our fellow traveler microbes are yet to be identified. Microbiology play a lot of role in identification of microbes and their use. So most of microbes can be found, the characteristics of microbiome maker, the kind of molecular techniques that could be used. Using DNA sequencing has been used, like 16S RNA have been used to differentiate various means of gene of various type of microorganisms. So bioinformatics method has been used for functional metagenomics, which represents an array of microorganisms in the environment that could play their role. We use them for isolation, making a copy sequence, and computational analysis. So what type of 16S RNA is useful for your microbiome study? You need to know, and you need to establish your own techniques that will give you the best goal. What might be the goal for human biome project? We need to develop a reference of sequence, provide information about disease, develop new techniques, and establish data, and a lot of other things. Scientists with human microbiome project have published numerous data. So you need to establish which type of human microbiome project do you think is important to your work, and a lot of other things. How might human microbiome project involve in fellow researchers like immunologists, molecular biologists, taxonomists, computer scientists, physical scientists, ethics, microbiologists, immunologists, ecologists, biochemists, and mathematicians, which shows a lot of other things. Now, can you imagine yourself from Pakistan and your institute winning a Nobel Prize very soon through the good work you are doing in your institute, especially on microbiome and gut microbiome? You need to discover such kind of award you can win. So you need to review human microbiome community around you, the kind of host, and a lot of other things. Scientists need to culture microbiome and another thing. Now I want to talk about fermented drinks. Fermented mix has provided an opportunity that ranges between a lot of opportunities, especially has become a current trend in the market from 2019 to 2016. So fermented drinks have become another of the, so the global meat production by species varies. You can see from cattle, buffalo, goat, sheep, camel, which shows that Africa also produce a lot of fermented drinks, and it has become a lot of things. So I will be talking about relevant microorganisms, health benefits, functional food potential, and how they could help us. Now, there are various regions in part of Africa that produces a lot of fermented drinks. Now, a lot of countries are suffering through a lot of economic problem. Now, the whole world is moving from linear economy to circular economy. Now, fermentation has become a process that will help us to enhance the level of gut microbiome that we have. So in Africa, there are more than 370 tribes in Nigeria alone, and they consume fermented drinks and a lot of it. So this shows a kind of fermenter that we use in Africa in production of fermented drinks. Not only that, interesting fact about fermented drinks from Africa shows that fermented drinks contain a lot of improvement and will enhance gut microbiome. So from the slide, you could see a lot of countries that produce these fermented drinks that will enhance our gut microbiome. Now, this shows our own fermenter that we use in Africa and locally produce. Example is Nunu and yogurt. This is where we produce our own traditional fermented yogurt. And you can see the genetic and the community variation and various kind of country from Africa, from camel, from cattle, from various tribes, how they produce their own fermented drinks, which could enhance.
Hello, hello, Professor Charles, can you hear me? Hello, hello, Professor Charles, can you hear us?
Hello, am I audible? Hello?
Charles, kindly share your screen, please.
Hello, am I audible?
Yeah, we can hear you now. You should share your screen yet again.
Okay.
Hello, am I audible now?
Yeah, it's totally fine. You can
Okay. So
proceed.
this shows the various sources of fermented drinks that we have in Africa. So also this shows the various type of fermented drinks. Now we have the various process we use in production of fermented drinks that could lead to improvement of God's microbiome. So microbiology of African fermented products has also been established. Most especially, we have lactic acid and yeast isolated from African fermented drinks. And this table shows various species of organisms and molecular techniques that has been used in characterization of this isolate from various parts of Africa, and has helped us a lot. Now, also, fermented and health application of fermented dairy products has also become important because of their nutritional composition. Like they serve as a good source of protein, and they produce a lot of motility capability, and they can enhance enzyme activity. And also, some of them form essential nutrients like calcium, linoleic acid, antioxidants, and a lot of other minerals like phosphorus, magnesium, and selenium. They can enhance the activity of the total protein, total fat, and lactose, and a lot of other things. And it varies from camel, goats, and a lot of fermented drinks products. They could serve as a functional food. They could serve as a active peptide in fermented drinks, and they produce all other ACE inhibitory and anti-thrombotic activity and provide a lot of bioactive components like immunopeptides, oligosaccharides, antimicrobial activity by working against pathogenic microorganisms. So some of these gut microbiome has found a lot of bioactive peptides, mostly from fermented dairy products, and they could serve as source of probiotics. So mechanism of action through which they work is modification of the gut, antagonizing excretion of pathogens through production of antimicrobial compounds, competition with pathogen bindings, stimulant, and a lot of other mechanisms. And also, in conclusion, I must tell you that we need to analyze seconds and investigate some of the standard techniques in order to check various fermented products that we have, and it's going to go a lot of way. So COVID-19 and fermented food products, some of these fermented drink products could help us to mitigate several kinds of COVID-19 disease or strains. On monomer of action is that some of these could suppress viral activity. Some of these gut microbiome have way of producing a macrophage that will engulf and improve the boosting. Some of these gut microbiome could increase the content of anti-inflammatory activity by preventing pro-inflammatory cytokine L1, IB, and transferrin and IL-6, and they could also form a kind of inhibitory against pro-inflammatory cytokine and a lot of other things. Now, I and Dr. Shakirat and a lot of other scientists around the globe have done a lot of work by tracing the probiotics present in bacterial species from buffalo, from Southeast Asia. These have gone a lot of way to help women microbiology to search for new probiotics, as we have talked about. And this work was done. We are able to show highest resistance was in NS3 against antibiotics like tetracycline and a lot of other things. So she shows the various growths of various strains. These are the growth pattern, and this is showing the homology of these strains that could produce probiotics. And this is showing the sensitivity test that has been established. This is showing the antibiotic resistance, and this is showing the enzymatic potential of these gut microbiome. And also, we also did a work together on isolation and characterization of cholesterol-lowering bacteria from buffalo and raw milk. And this is showing that probiotics could be derived from animal source and serve as a source of human health benefit for both animal and human. So interestingly, we discovered that they could produce a lot of health benefit, like lowering of cholesterol. And this showing the morphology, this showing the homology, this showing the various pH, and this showing the various sorts activity, and this showing the antimicrobial activity, antimicrobial response, and this showing the enzyme activity, and this shows the translation of the cholesterol lowering activity, and this showing the environmental effects. And also, gut microbiome are not only producing probiotics or biotics alone. There are a lot of work I and my research group have done. They could serve as agricultural and biotechnological application. They could serve as biotechnology for rumen, which they could use to produce a lot of activity like biogas. Some of them could be used in production of enzymes like cellulose that has a lot of wider industrial application in textile. They also use them in pharmaceutical industry. We have also established that some of these novel organisms are involved in production of biogas, and they could be applied in production of commercial animal feed. Also, they play a lot of role in digestion and enhancing the digestibility of active food in animal. Now, on this note, I would like to encourage all of us to involve in a kind of teamwork and interdisciplinary approach. We need us to get our gut microbiome, and I want to sincerely appreciate your university and your research institutes. You are doing a lot of wonderful work in terms of gut microbiome, and we need to still collaborate more, work together, and form a kind of team. So on this note, I want to thank a lot of country, a lot of university that has invited me as a visiting professor to come and have a talk. And yearly we have a biotechnology program. I would like to invite you I've also consult for the government of Nigeria in order to advise them on how to use some of these strains and in improving the sustainability. Some of the memory and a good memory, and I would like to thank some of my hosts that, and I also invite you to join some of my editorial group as where I serve as the series editor and send your book. And very soon, I and your institute are also going to publish a book under review, and which will be published very soon. So special thank, I would like to thank Dr. Shakirat for inviting me. And I want to thank your research group also for bringing me to talk to all of you in this conference as a distinguished professor of eminence. So feel free to get in touch with me, and I'm looking forward to collaborating with all of you. Thank you very much. I am very grateful.
Thank you so much, Dr. Charles. It's indeed our pleasure, and Dr. Chakra is very thankful for you to be part of this conference. Moreover, there is a question from the audience. Can you hear me?
Okay, I can hear you. I can hear you. I'm with you.
Okay. There is a brief question from the audience that says that what about the gut of an animal that is just recently born? Does that gut contain any microbiome or any microbiota?
Yeah, research has shown that definitely there are presence of microbiome present there, and we need to intensify on the application of omics, the application of genomics, proteomics, and bioinformatics and metabolomics in order to establish the role some of these new microbiome from this newborn could play, or they could serve. It's going to play a long role for us to know their function in terms of ecology contents, earthwise, and a lot of all functions.
Okay, thank you so much. Thank you so much, Dr. Charles. It's indeed our pleasure, and we hope to see you again very soon, inshallah. Thank you.